Welcome to today's seminar, everybody. I will give it a few seconds for everybody to attend. There are still people joining, so we'll wait a few more seconds. Okay, great. Welcome everybody to today's Education Committee seminar series. I'm very happy to be hosting this Trainee Talk session today. Before we start with the seminar, I would like to make a couple of announcements on behalf of the Cachexia Society. We have a new mentoring program, an initiative to feature trainee members of the Cachexia Society. This is meant to improve the visibility of trainee members of our society, and it's also meant to get to know these early career scientists. So if you follow this link on the screen or just go to our website, then you will find the CVs and projects of some of our trainee members. If you find this a cool initiative, if you would also like to be featured, please contact our admin team, and they will be happy to assist you with that. Another thing the Education Committee does for fostering this mentoring of trainees is to initiate these trainee talks, as we have one today. This is really meant to highlight, to put you on stage, put your research on stage, and to show off your results. This is on a nomination, self-nomination basis, or you can get nominated as well. The next trainee talk session is in September, and we have already selected 3 speakers. But there will be another round of nominations coming around the end of the year. Then a new initiative that I wanted to share with you, and you might have seen an email by our president, Andy Judge, is that we have now introduced office hours with board members. This is meant to provide mentoring opportunities to our trainee members. You can book an appointment with members of the board or members of the Education Committee. And this is a members-only event. So only if you are a member of the Cachexia Society, you can sign up for these mentoring initiatives. So please consider signing up. It's very free for trainees. And another thing you might want to put in your calendar already is our upcoming Cachexia conference, which will take place in Turin, in Italy. And it will take place in the end of September next year. On May 15th, we will have our next speaker, Dr. Anne-Catherine Morin from France, and she will talk about the GCN2, EIF2 alpha, ATF4 signaling pathway, and essential amino acids. So you're very much invited to join this seminar as well. Now, today, it's my great pleasure to introduce these 3, or I should say 4 speakers. We will have Kandy Velazquez from the University of South Carolina, speaking about exploring the impact of the immune system on cancer cachexia. We will then have Brittany and Aneesha Dasgupta introducing their new initiative on women in cachexia. That is a new initiative hosted by the Cachexia Society. And lastly, we will have Sarah Santiloni Cury from the Sao Paulo State University in Brazil, speaking about the human cancer secretome and single-cell resolution. So we are very excited to host all of these speakers. Welcome once again to everybody. And I'll leave the screen now to Kandy.
Hi, everyone, and thank you for the invitation. I'm gonna share my screen. Can everyone see it?
Yes.
Yes.
Thank you. Well, I'm going to be talking about my interest, that is going to be how the immune cells are affecting cancer cachexia. The reason why I select the immune cells is because the loss in lean mass have been associated with an increase in pro-inflammatory cytokines. And these pro-inflammatory cytokines can lead to the release of acute phase response proteins from the liver. And this can by itself lead also to metabolic disturbance, such as increase in energy expenditure and decrease in energy intake or anorexia that have been shown as well. Despite attempts to improve muscle loss and weakness, we have seen through parental and enteral nutrition, we have seen that hasn't been as successful as people will like, and the energy expenditure in colorectal cancer or also other types of gastric cancers have been shown to be modulated, like in pancreatic cancer. Therefore, because I study colorectal cancer, we decided to go with the immune cells instead, the loss of or decrease in energy expenditure. The immune cells that I'm interested in are macrophages and platelets. Reason why I select them is because it has been shown that there's an increase in a marker of macrophages, CD68, in patients with cancer cachexia, pancreatic cancer cachexia, as it can be observed in this figure here, in this graph. Additionally, it has been observed that an increase in platelet and a decrease in albumin has been shown in patients with pancreatic cancer. And this decrease in albumin and increase in platelets have been associated with muscle wasting as well as poor survival. Thus, to explore these immune cells or these cellular events, I selected the APC in mouse. This is a model of colorectal cancer, and is due to a mutation in the tumor suppressor gene, the APC gene, of Adenomatosis polyposis coli. And in this model, you're going to have intestinal polyps that are going to start growing around 4 weeks of age, and anemia, muscle weakness, and muscle loss are going to occur during a period of 20 weeks. Here I have a table of Dr. Bonetto that illustrate the different models of colorectal cancer cachexia, so the audience have an idea how these APC means relate to them. There is usually a bigger loss of muscle mass. So in my experiments, we have wild type or C57BL6 that are leader main control. We have precachectic mice, so these mice are going to have 4% or less body weight loss, and the severely cachectic mice are going to have more than 10% body weight loss at the time of that we sack them. So we observed that, as expected, we have that the severely cachectic mice have a decreased percent body weight and also have a decrease in muscles of the limbs, like the plantaris, gastrocnemius, and quadriceps. And we also observed the TA, by the way. And when we look at the blood, whole blood, we have found that there's no difference in white blood cells, but there's going to be a reduction on lymphocytes and an increase of neutrophils that also have been shown in humans. And here we have the neutrophil to lymphocyte ratio, and there's an increase in here, as expected. And in platelets, we have an increase in platelets at the precachectic stage, and we have an increase in platelet activation, that is the mean platelet volume, at severely cachectic stage. So this was the basis of my R21 to study what is the role of these platelets. The R21 was just funded in March, so I don't have data for that, but hopefully I have something to bring to the Cachexia, Keystone Cachexia conference in 2 weeks or 3 weeks, I'm not sure. And but we are working on this right now in the laboratory. Some interesting fact is that in a study in Canada, that they study their population because they have a lot of blood count data. They saw that an increase in platelets was found up to 3 years before cancer was observed in patients with gastric cancer, colorectal cancer, and ovarian cancer, that all of them lead to or can lead to wasting. We utilize other models, the C26 and also the LLC, and we observed similar effects. Other groups have shown, by the way, already that there's an increase in platelet number in CD66, CD66 tumor-bearing mice, and we also observed that there's an increase in the activation of platelets on LLC mice, Lewis lung carcinoma. We have that there's an increase in markers of neutrophils in severely cachectic mice, also an increase in M2 macrophages in the skeletal muscle of severely cachectic epistemine. There's an increase in anti-inflammatory cytokines like IL-10. And in precachectic mice, there's going to be an increase in IL-1 beta that it's maybe related to the platelets, and that's something that we are going to be following up. For the pathways of the TGF-beta, because others have also shown that fibrosis is involved in the later stage of cachexia, skeletal muscle wasting. We observe that there's an increase, not significantly different, of TGF-beta1. This is gene expression in the skeletal muscle in the gastrocnemius, but we observe an increase in TGF-beta2 and also TGF-beta3. And it seems that it might be through the canonical pathway of TGF-beta with SMAD3 being upregulated. And we observe fibrinogen also that is upregulated in precachectic stages. And for us, this is important because we think that if we can find something at different stages during the precachectic, cachectic, and severely cachectic stages, maybe we can find a way to treat the disease at different time points, because everyone comes or realize that they may have cachexia at different time points. So this is my hypothesis. I believe that platelets are the main are playing a main role at the beginning of the disease during precachexia, and this is increasing the number of platelets, and then later on they will going to be increasing their activation, and this is maybe because the platelet decreased due to the treatment or other factors. We believe that platelets are involved in the TGF-beta activation. That's something that we're going to study. We don't know how yet, but we have our other hypotheses as well. We have that TGF-beta1, we believe that is increasing the macrophages, especially the M2 macrophages, and this is what is helping to promote the fibrosis. So I have like a 5 to 10 years plan of studying, and hopefully we have good papers after that. So other thing so part of the data that I'm going to show you today is something that my student just have some pilot data, and we have in here that we are injecting liposomes with clodronate. When the clodronate, what it's going to do once the macrophage ingested, they are going to be released in the macrophage, in the cytoplasm, and then apoptosis is going to occur. So we're going to be depleting macrophages. This is not obviously the best model to deplete macrophages, but it's one that is available, and it's going to be depleting macrophages everywhere. But we're using it only twice a week for 2 weeks only, so it's very short term. And what we observe is that this is in cachectic mice, more than 10% body weight loss of the epistemine, and we observe that there's when we give them liposome with encapsule, that means with PBS, we have a decrease in macrophages. So in this case, the macrophages should be intact because there's nothing in this liposome, it's only PBS. So once it's released, they're still alive. the macrophages, but when we give the liposome with clodronate, there's going to be depleting macrophages, and it seems that it's not as reduce the loss of muscle mass in the gastrocnemius and also in the quadricep muscle. When we observe or when we look for function, we see that there's it seems that there's an improvement in muscle function when we compare APC-mean mice between themselves. And also when we compare them in the RORA that to measure neuromuscular function. So we think that it's great to see that macrophages, like an acute or a short impact, has these drastic effects in mice. So I want to thank everyone in my lab who have been doing this work and the people who fund us. Thank you so much.
Thank you for this excellent talk, Kandy. The floor is now open for discussion. If you have a question, please type it into the chat, into the Q&A. If you would prefer to ask your question directly, please raise your hand and I can try and unmute you. This is also possible. Now, while people are typing, Kandy, can I ask you, why do you think, I found it very intriguing that platelets come up in patients before cancer is detected. Can you speculate on why that could be? Because that would be a very interesting way to help with the diagnosis.
Yes, my speculation is that because it's due to anemia, because of the low in iron. Mm-hmm. But I'm not 100% sure. Like this obviously will have to be investigated, but that would be my best guess right now.
Okay, very interesting. Let me see. There's one question in the chat by Niklas Colau. Really nice work, Kandy. Besides the measurement of muscle weight, did you perform analysis of muscle cross-sectional area? And did clodronate affect other cell types?
Yes, I did cross-sectional area, and there's also an improvement in the ones with clodronate. For if I check other immune cells, you mean like dendritic cells? Like we didn't run flow cytometry in this particular set of animals. So, but we know that neutrophils and dendritic cells, anything that phagocyte will be eating, that can eat the clodronate. Yes, we did measure tumor or polyp number, and there was no difference in polyp number or polyp size.
And would there be a possibility to deplete, for example, M2 macrophages specifically?
This particular clodronate is monocylated clodronate. So usually the M2s are the one who have the monocyllate receptor. So it's more probably that they will be targeting, but the macrophages are so pliable and they can change their phenotype. So I couldn't say that if they are in between the transition from M1 to M2, it might not be targeting them as well.
Okay. Teresa Zimmers is asking, did the clodronate reduce tumor burden or tumor histology or gene expression at all?
Sorry. We are aware, no, it didn't change tumor volume or the gene expression. I haven't measured gene expression in the tumors, but that's something that we can definitely do.
I think that would be interesting to see tumor progression effects or similar. Okay, then Nikita Ballerau is asking, did you see any effect on adipose tissue?
Well, in our APC-mean mouse, for adipose tissue, it goes very early, and at this point that it's usually over 18 to 20 weeks of age already, they don't have adipose tissue. So we didn't see any difference of promoting fat storage in our
Okay, great. And then the last question is by Fabio Penna. platelets produce huge amounts of extracellular vesicles. Have you tried to measure the amount and the content released in healthy versus APC min mice?
I haven't. That's in the plan of measure. Yes, we have also planning to measure if the platelets in the APC min mice are aggregated to neutrophils or are having aggregations to monocytes. So we have a lot to do.
Okay. Okay, so it's not the last question. Actually, two more came in. Maybe short answers to those. We have the question if you performed flow for immune cells from the muscle.
We do it in the lab, but in this cohort that I show, I haven't. But that's coming.
Okay, great. And then the last one is by Andrea Garcia Ray, who has questions related to the force test. How did you measure the grip strength in the animals, and did you perform measures at different time points during the experiment?
We performed 2 measurements before the treatment and after the treatment. We didn't do multiple, so it's only 2, pre and post. And what was the question about how I did it? We put the 4 pause.
Yes. 4, okay.
Yeah, it's 4, yes, the 4 time.
Okay. Excellent. Thank you so much once again for your talk and for the very nice discussion.
Thank you for the invitation.
Glad to have you here. Now, the next speaker of today is Aneesha Dasgupta, and he will share the stage with Brittany Counts, I think, who will come on stage later, introducing the Women in Cachexia initiative.
Good afternoon, everyone. I'm just going to share my screen. On behalf of WCX, I want to thank everyone for giving us the opportunity, especially the Cachexia Society and the Education Committee. So Brittany and I are here briefly just to discuss our newest initiative and what we plan to do and the help that we need from the community. So why we wanted to start this initiative was because I don't think it's a surprise that the share of women in STEM workforce is significantly lower than those in non-STEM workforce. In 2015, it was about 28%, and in the last decade, it has only raised to about like 5% more. And here we are showing the data on women because the data on other historically underrepresented gender is not there at all in the literature. And we also see that women do enter the workforce at the entry level in STEM around 28%, but as with each promotion, the percentage keeps decreasing. At the highest position, they are at like 12% of the workforce. So this is mainly why we wanted to start this initiative, because it has been known that women's professional organizations develop women's leadership capabilities, foster relationships, and generate networks. And we want to foster all the amazing women and other historically underrepresented like individuals in the community to come together, and we have some aims for that. So briefly, I would like to say that WCX is under the DEI committee of the Cachexia Society. So we do, and the DEI committee is headed by Dr. Teresa Zimmers and Dr. Maria Sealander. So we do work with them in fostering our goals. So the goals of WCX are twofold. The first is to introduce discussions about and promote gender equity among Cachexia researchers. And the second aim is to increase leadership capacity, sense of belonging, and networking and mentoring among women in Cachexia field. I would again like to remind you that women here also includes the historically marginalized communities, genders. So with this, I would like to give it to Brittany.
Thank you, Aneesha. So to address our 2 aims, for aim 1, as Aneesha just said, is to introduce discussions and promote equity amongst the cancer researchers 2 ways. So first, we will have stories and science seminars, and all this will be on the webpage. So if you want to find out a little bit more specifics about each one, please go there. And then we'll also have the Women in Cachexia seminars. So those will be research talks given by individuals of marginalized genders in the Cachexia research. And this will hopefully promote and discuss their research along with maybe other endeavors that they have encountered along the way. And then for aim 2 is to increase leadership and a sense of belonging amongst the Cachexia community for women in Cachexia. So this will be done through the Women in Cachexia Initiative Forums. So these are open discussion forums that we will have. You can discuss your career, you can discuss leadership positions, and mentoring opportunities, obstacles, anything basically that you want to discuss. We are here to listen and promote and help as much as we can. And then the last part of aim 2 is to have the network. So we want to have a place that you can go to find support or ask for support from other Cachexia researchers in the Women in Cachexia initiative. So we do ask that all of this, you can support and give your comments as much as you want, because we want to hear from you. And then so far, we are very optimistic and excited about the community so far, because we've had a lot of support. So far, we've had 44 survey respondents. And what we thought was really promising and hopeful is that most of that was from investigators. And we do want to point out that if you are an investigator, if you could share this link that we will put into the chat with your mentees, that would be super helpful, because we want to, there's not so many mentees actually that can get some of the emails. So we want to give the opportunity to them. And it is very exciting that we've had a lot of interest around the globe, and we hope to keep this going with the next link that we send out soon. So as I've already said, we really want to hear from you, because we want to make this a community that is all involved. So we want everyone to have an input. So we will put the link in the chat, and if you could just take, it's very, very fast. So if you could just take the time to fill it out, we would appreciate that. We will have another survey to follow to see what everybody wants. We don't want to give you too many things to do, but we want to be able to be a supportive community for everybody. So please fill that out so we can make this the best that we possibly can. And then we want to thank everybody for all that they've done so far. The Society, thank you very much for allowing us to start this initiative. And so far, this is the founding board. We will all be at the Keystone Symposia in 2.5 weeks. So please come see us there. We want to discuss and meet up. And if you have any questions or concerns, please reach out to any of us. And with that, we'll have, if anybody has questions or comments.
Thank you for having us here.
Excellent. Thank you so much. Thank you so much, Aneesha and Brittany. I don't know about you. I had some problems with the connection at the last talk, so apologies if I didn't get all of it. But I would like to congratulate you for this initiative. I think it's a great idea, and it really deserves all the support that we can give you. You know, this is very inclusive. So please, everybody is invited to participate, to give opinions. Yeah, congratulations for this. Actually, there's a lot of applause and congratulations also in the chat. So this is very good to see. Would anyone have questions for Aneesha or Brittany? There's one question actually. Is the survey link on the CCS website?
I put the link in the chat, but we will double check to make sure the link.
Yeah.
We'll double check.
I think there's another question. When will the seminar stock launch within this program?
Yeah, it just came in, exactly.
So we are working on it. I think, sorry, Brittany. We are working on it. So I think the first one, which will be after the Keystone, and we will send out a doodle poll for it too. And it would be the WCX committee talks, right? The seminar, which is to discuss obstacles and stories And then I think we will start with the DEI seminar this summer.
And we will say in the polls, if you can put your preferred time zone, that way we can accommodate as many people as possible.
That's great, thank you. Let me check, please, if I have some hands up here in the attendees. Okay. No, or apologies if I've missed them. I don't see any. I see a lot of, clapping hands. So I think this is great and shows the general support. And apparently there are no immediate urgent questions that people want to ask now. But thank you very much for introducing this, and good luck with this, I will see you in Keystone and potentially many of the attendees today. So there will be a lot more room to discuss this in the future.
Thank you.
Thank you.
Thank you. Excellent. So our last speaker of today is Sarah Santiloni Cury. Please, Sarah, you're ready to go.
Yes, let me share my screen. Just 1 second. Okay, is that good?
Yes.
Okay, so good morning, good afternoon, everyone. It's an honor to be here today to talk about some results of my postdoctoral project that I developed here in Brazil, in São Paulo University, under the supervision of Professor Robson Carvalho. And we know that the cancer secretome plays an important role in cancer cachexia. So the tumor produces and secretes a lot of factors, such as pro-inflammatory cytokines, such as TNF-alpha, IL-6. And these secreted factors induces a lot of different alterations and wasting in target tissues. So we know that the tumor secretome is a source of cachexia-inducing factors. But the cancer types don't have the same prevalence of cachexia. We know that pancreatic cancer and gastrointestinal cancers have the highest cachexia prevalence, and patients also present the highest weight loss, but patients with prostate cancer and breast cancer is the opposite effect. We don't see so much prevalence of cachexia and weight loss. So we previously investigated why these cancer types different in cachexia prevalence, and in this paper that was developed by Paulo and Jason, we found that the cancers with highest cachexia prevalence, they present also higher levels of cachexia-inducing factors. They upregulated more number of cachexia-inducing factors. So, for example, pancreatic cancer, this first column here in the heat map, demonstrates that the majority of cancer cachexia-inducing factors are upregulated, and cancers such as breast cancer and prostate cancer, they don't change the gene expression, or even they downregulate some factors. And we also saw that the cancers with low purity, here is only pancreatic cancers. These cancers with low purity, they also present higher levels of gene expression of cachexia-inducing factors, suggesting an important role of tumor microenvironment for this production. And these review demonstrated that cells within the tumor microenvironment, they contribute to the expression of IL-6, TNF, and other factors. So now we have the single cell technology that can help us to better understand the tumor microenvironment. So different from the bulk RNA sequence data, which is the collection of cells that are processing together, and we only have an average expression of all the cell types that compose the tumor. We now can see with the single cell technology the gene expression of each of the cells that are in the tumor microenvironment. So our aim was to characterize the transcriptional profile of the tumor secretomes in single cells in a pan-cancer analysis of tumor types with different cachexia prevalences. So to do this, we use publicly available data sets, single cell data sets. This public data set is called 3CA, and they performed uniformly pre-process of the data, so making every single data comparable. So we use this data. And we finally went up by analyzing 10 cancer types and almost 1 million single cells in 36 different data sets. So these ones are highlighted here are the cancer types that we analyzed. And we also follow the pipelines for single cell analysis that are suggested by CIRAT. So we performed the clustering, identification, the cell type classification, and we also evaluated the cancer secretome by using a predefined list of genes that encode for secreted proteins. So we use this list for further analysis. So some of our results. First, we select 7 cell types that are commonly found in the 10 cancer types that we selected. So we included for further analysis the malignant cells, T cells, macrophage, fibroblasts, B cells, endothelial cells, and dendritic cells. And we evaluated whether the percentage of these cells within the microenvironment would be correlated with the cachexia prevalence, but we haven't identified any correlation. But here we can see for the immune fraction and also for the stromal vascular fraction, we can see 4 cancer types that have the same trend. And interestingly, these cancer types are pancreatic cancer, colorectal, lung, and head and neck cancer. And they have the opposite direction in immune fraction and stromal vascular fraction. So we applied principal component analysis to see if the secretome genes that we are investigated could cluster the cell types differently. But we can see that the expression profile of the cells are very similar despite the tumor type, the cancer type. But here we can see that the fibroblasts present a higher variation in the gene expression of the secretome genes, suggesting that this cell has a more dynamic role in the tumor microenvironment depending on the cancer type. Next, we've evaluated the expression profile, and we can see here in this heat map that the cells, they present an expression profile that is specific of this cell type. So here we have, as an example, the genes that are specifically increased in B cells, and we also saw that the function of these genes were correlated with the cell types that we saw the enrichment. So here we can see enrichment of these genes in the cell type, and we also saw that these genes are associated with the positive regulation of T-cell chemotaxis. And by doing this, we can set the transcriptional profile of each cell type within the tumor microenvironment. But we also performed differential expression analysis to see if the upregulation of secretome genes are associated with cancer cachexia. And we found that the upregulation of secretome genes in malignant cells and fibroblasts are correlated with cachexia prevalence. And we also identified that the combined cells also upregulated secretome genes that are correlated with cachexia prevalence. So we confirmed our previous findings in TCGA, but now displaying a more important role of malignant cells and fibroblasts to increase the production of secreted factors. And what about the cachexia-inducing factors? We expanded our analysis, so this was a PhD project developed by Victoria, and she went to literature and some mining tools, and we selected 123 genes that are associated with cachexia and already published in the literature. And here we can see that the fibroblasts, the macrophages, and the endothelial cells upregulated the highest number of cachexia-inducing factors, demonstrating that these cells are playing an important role in the expression and production of cachexia-inducing factors in the tumor microenvironment. And here is the landscape of these cancer-associated these cachexia-inducing factors, and we can see that as the secretome, we have a profile specific to the cell type and not the cancer type. So here we can see some genes, cachexia-inducing factors that are mainly expressed by fibroblasts, and they are associated with inflammatory response, AGF1, AKT signaling, and so on. So now we have an overview of these cachexia-inducing factors in each cell type, in each cancer type. So after doing this analysis, we went to see how this secretome is contributing to the communication between cells in the tumor microenvironment. So here we can see that the pancreatic cancer, which is this blue chart, we can see that a lot, most of communication pathways are altered in pancreatic cancer. While in breast cancer, we only see few communication pathways altered by this communication. We also saw that the number of interaction increases with the prevalence of cachexia in malignant cells, macrophages, and also fibroblasts. And we also identified that the strains of interaction, the most interactions occur between the stromal cells, which is the fibroblast and the endothelial cells, with the malignant cells. So they present a higher weight of interaction in all cancer types that we investigated. So we went to the fibroblasts and malignant cell communication, and we found these communications there we can see a higher correlation with cancer cachexia. So when these communications increases, or the probability of this communication increases, we also increase the prevalence of cancer cachexia in the tumor type. And here the interesting thing is that cytokine 4 is the main receptor of malignant cells that are receiving communication signaling from the fibroblasts, so suggesting that these cytokine 4 is important for cachexia. So some of our conclusion is that the secretome has a cell-specific expression profile in human cancers. The number of upregulated secretome genes in fibroblasts and malignant cells correlated with cachexia prevalence. The fibroblasts and macrophages increase the expression of the highest number of cachexia-inducing factors, and we also saw an elevated cachexia prevalence that correlates with increased interaction within the tumor microenvironment mediated by the secretome. And we also highlighted that cell communication indicated the essential role of fibroblast secretomes in cancers with high prevalence of cachexia. So we want to demonstrate that these data will be available in this database that we're working to publish and open to all the community to analyze some data. So we are planning to include this single cell RNA sequencing data in this cancer cachexia omics database, also called CacauDB, that we're collaborating with Dr. Teresa Zimmers to make it available soon as possible. So I would like to thank you for your attention, and I'm happy to receive your questions.
Thank you, Sarah, for this fascinating talk and for this really interesting approach and new approach to better understand cachexia and the role of the tumor for the disease. Okay, I don't have questions yet. So, oh yes, I do actually. I have 2 hands up, so I will allow Fabio to talk. Let me see if that works.
Okay.
Yes, should work. Fabio?
Hello, can you hear me?
Yes.
Hi, Maria. Hi, Sarah. Nice talk. So I prefer talking rather than writing because it's not an easy game you try to play. So you highlighted the role of fibroblasts, but we're talking about cancer-associated fibroblasts. My question is, since we are interested in the immune cells, in the muscle, in liver, in all of those tissues in which we do observe the phenotype, have you thought of the role of fibroblasts in other tissues? Because fibroblasts are highly abundant everywhere. They do receive signal. They are highly activated upon inflammation and so on. So are you gonna follow up with studying fibroblasts around the body?
Yes, we are now working on RNA sequencing data that we extracted a lot of different tissues from LLC model. And we can do that we call digital cytometry, which is using the RNA sequencing data to infer how much of cells and fibroblasts or other cells are in this tissue. So we are working on this now, and I think we'll probably have some interesting findings about muscle. We are also analyzing adipose tissue, visceral adipose tissue, heart, liver, and of course the tumor.
Fantastic. Thank you.
Thank you, Fabio.
Mm-hmm. Maybe along that same line, Nikita Balarao is asking if these CIFs are already functionally validated elsewhere to cause cachexia.
Yeah, we are working on some projects to validate this finding, but it's quite difficult to have patient data that we know for sure in public databases that we know for sure they are cachectic. So our main limitation is that because we have the tumor data of these patients, but we don't have the clinical information about the weight loss and muscle function, nothing like this. But we have several published data sets that we can search, and we have this information about the clinical status of this patient.
Mm-hmm. Next, I have asked Robson Carvalho to ask a question in person. Can you unmute? No. Okay. Sorry, there was another question by Bilgun Erkem Ocher, but now I cannot see. Ah, yes, there you are. Okay. So, Bilgun.
Hello.
Hello, we hear
Okay.
you.
Nice talk, nice project, enormous work. I have to congratulate. So I was just working. Do you have any plan to investigate some proteomic profiles? Is there any way that you have access to some patient samples or mouse models to integrate into this database?
Yeah, we don't have patient data, but we have proteomic data of this cancer model that I told you. But we only have the plasma samples, so we don't have the proteomic profile of each individual tissue. We don't have that.
I see. So in terms of human proteomic and metabolomics, I think you probably know the TracerX project that is in UK. So maybe you could contact with them if they're willing to collaborate or something, because I saw they have presented some proteomic data, preliminary, and some metabolomic signatures as well. So it's enormous. Thank you.
Okay, thank you. It's a really nice idea. Thank you.
Then we have another few questions in the Q&A. Mauricio Beriel Diaz is asking, well, he congratulates you for the great project. And he is asking, how detailed was the information on the presence or absence of cachexia related to the data sets? And did you introduce any measures to enable the comparability of the cachexia phenotypes?
Yeah, we don't have this information of the single cell data. We don't have. The only thing that we have is the prevalence, that we can make correlations and inferences. But we have public data that we are currently analyzing, but it's only for pancreatic cancer patients. And for this data set, we have the gene expression profile of the cancer, and we also have the tomography, the patient's tomography, to make muscle area analysis and to confirm if they are cachectic or not, and to see if these secretome genes are really there and differ from patients that present higher muscle mass or low muscle mass. We are working on that now.
Mm-hmm. Great, thank you. And then the last question I have here in the Q&A is by Laure Binders, who asks if these CIFs, by definition, are common to all the cancer types that were studied.
Yes, they are, but they don't present the same expression level, but we can identify all these genes at some level in single cell resolution. So that's why single cell is really interesting, because we can see even a small amount of gene expression, but we can identify these on that cell types, even if they are rare cell types.
Mm-hmm. Excellent, thank you. So I don't see any more raised hands or questions in the Q&A. Yes. So this means we are through with all the questions. So once again, Sarah, thank you so much for this really presenting this really exciting project. I would like to thank all the speakers once again for being here, for making this a wonderful session. And thanks so much, and looking forward to seeing many of you once again next month on the 15th of May. Thank you.
Thank you. Bye-bye.